Thank you for joining today's IAFP webinar, Cronobacter in the Spotlight, New Insights into a Known Organism. Today's webinar was organized by ILSI Europe with the support of IAFP's PDGs, the Low Water Activity Foods PDG, Microbial Modeling and Risk Analysis PDG, and the International Food Protection Issues PDG. Today's moderator will be Annette Winkler from Cargill, Germany, and all webinars are sponsored by the IAFP Foundation. Before we begin, there's some housekeeping items to remember. It's important to note that all opinions and statements are those of the individual making the presentation and not necessarily the opinion or view of IAFP. All attendees are muted. Questions should be submitted to the presenters during the presentation via the questions section at the right of the screen, and questions will be answered at the end of the presentations. This webinar is being recorded and will be available for access by IAFP members at www.foodprotection.org within 1 week. And at this time, I'm going to turn the webinar over to our representative from ILSI, Isabel, and she will give a short presentation on ILSI Europe.
Thank you, Dina. Providing science when we need it, that was the intention behind this webinar. My name is Isabel Gunning, Scientific Program Director at ILSI Europe. So thank you for IAFP to supporting us with this webinar. And for those who don't know ILSI Europe, which stands for International Life Sciences, start with creating collaborations. And the first collaboration, for example, is the Microbiological Food Safety Task Force of ILSI Europe. And you can see here the screen, the composition. So we have at least industry members, at least 4 or 5 of them, provided by the scientific advisor from Wageningen University. And we also collaborate a lot with Camden BRR, and you have the experts here today, Roy, with us. Of course, also collaboration extends beyond this webinar with IAFP. Also in our expert groups, we also have this collaborative approach, as you can see on the screen. Once a collaboration is established, we identify common pre-competitive challenges, and the Microbiological Food Safety Task Force investigates microbiological issues in food that relate to public health risk and facilitates the development of harmonized science-based approaches to predict and prevent microbiological risks, so much needed nowadays. Then we assemble the science, and all of our science is open access available. Here you're seeing some extracts from the publications, but you can also find them, all of them, on the website. And besides providing the science, we communicate and disseminate, like today in this webinar, but also at the IAFP symposia. We have sometimes frequently sessions, webinars, and also ILSI Europe sessions. And with this, I end the ILSI Europe show, so as we said, and I will hand over the words to Annette Winkler, also chair of the Microbiological Food Safety Task Force from ILSI Europe. Annette, over to you, and please enjoy the webinar. I hope it's interesting for you.
Thank you for the nice introduction, Isabel. And with that, I think I don't spend time on this slide. You have time to read anyway. It's more important to know who are the panelists today and who are going to answer your questions. So the first one I want to introduce is Seamus Fanning. He's working at the University College Dublin at the UCD and is currently the director of the UCD Center for Food Safety, with more than 30 years research experience and extensive research experience on Cronobacter. In that respect, and also in others, he served as an expert member on several WHO FAO missions, and he's also an elected fellow of American Academy of Microbiology since 2019, and a serving member on several editorial boards of journals, including the Journal of Food Protection. And Seamus will give us a short introduction to the topic after the general introduction of all panelists. So with that, then moving to the next panelist, François Bourdichon. He has also an amazing experience in the food industry, more than 15 years, and since January 17, he's a principal consultant at Food Safety Microbiology and Hygiene and a research collaborator in DISTAS. I don't dare to pronounce the Italian names, but it's a university in Italy. He's a member of IFP since 2007 and also an elected fellow American Academy of Microbiology since 2019. He did a lot of work with the industry, also in troubleshooting several cases. So he also brings in experience in the field. Then going further, our next expert here for this roundtable is Marcel Swietering. He is since 19 years professor at the Wageningen University in food microbiology. He has also industry experience coming from Danone, where he worked for 5 years. He's the chair of ICMSF and active in ILSE Europe in the microbiological food safety task force and very active in IFP as well. Marcel has a lot of experience when it comes to risk assessments, where he works a lot also with industry. Then our next panelist is Roy Betts. Roy Betts works at Camden BRI in the UK. He's currently the head of microbiology. Started really as managing a research team at Camden BRI, concentrating on research, development, and validation of microbiological test methods. However, over the years, his interest moved to the assessment of the microbiological quality safety of foods, advising industry on techniques, procedures to produce and market high quality and safe foods. He's also an active member of ILSE, of the UK Food and Drink Federation, Hygiene Subcommittee, and the UK Advisory Committee on the microbiological safety of foods, as well as the British Standard Institute and ISO committees dealing with microbiological test methods. Then our next panelist is Stephen Forsyth. It's a pleasure to introduce him. He's a retired professor, but still very active, I should say, from the Nottingham Trent University, because currently he's a guest lecturer at universities all around the world, from Finland to South Africa. And previously he was the external advisor to the UK's Food Standard Agency, European Food Safety Authorities at EFSA, and the WHO. In that respect, he served as an advisor on all 3 FAO WHO risk assessments on Cronobacter in infant formula and ready-to-eat infant foods in famine relief, with a lot of publications on Cronobacter and related organisms. So I think we have quite an excellent round of panelists, and with that, here's a reminder: when you have questions, please ask them in the question box, not in the comment chat box. Thank you for doing that, and I'd like to hand over then to Seamus for an introduction to Cronobacter species. Thank you.
Thank you very much, Annette, for the kind introduction, and also to ILSE and to IAFP for the invitation to just present some background thoughts on the bacterium that's at issue here. So my purpose in the presentation is just to try to give you an overview of Cronobacter, bearing in mind the reason why we're meeting here on this seminar webinar at the moment, because of the ongoing issues that we see in the public domain. So just to kick off, this is a bacterium, as you all know. It's a member of the Enterobacteriaceae family, which also includes similar organisms like, for example, Salmonella and E. coli. Therefore, it's gram-negative. And so it espouses many of the phenotypic behaviors and traits of gram-negative organisms. It was designated in 1980 as an Enterobacter sakazaki, and the taxonomy was revised some years later in the mid-2000s, where this new genus was created and became known as Cronobacter species. It's a very easy bacterium to handle in the laboratory. It's easy to grow it, easy to culture it. And one of its most characteristic features, I think, and I'm sure several of the people will pick up on this during the course of our discussion, is its desiccation tolerance. So this is perhaps one of the most important phenotypes that this particular bacterium expresses. And so trying to understand that phenotype is very relevant towards trying to come up with strategies to improve controls. It is considered as a rare opportunistic pathogen causing nosocomial infections, and I'll pick up on that again in the coming slides. So in terms of what we know epidemiologically about this bacterium, we probably didn't know a lot about it until very recently when we started to really recognize it. And so in my slide here on the top part, you see a time-related distribution of the numbers of cases that were being reported in the US, and you can see that these blocks are starting to increase more frequently as you go to the right-hand side of my graph, which suggests that we are recognizing more and more these cases and outbreaks when they occur. And also on the bottom of my slide, you see recorded here just a number of the global cases and outbreaks that have been reported over the last number of years. And the one that I've marked in yellow, of course, is the one that's currently in our sites. The reason it's in our sites is because it was reported back in February of this year that there was a contamination of a powdered infant formula batch which was manufactured in the United States and which gave rise to 4 cases of illness, 2 of which died. There were a number of the US states involved where this organism was detected in powdered infant formula. And this, of course, initiated a recall for the company involved, and then the pursuant investigation, which, as I understand it, is still ongoing. And it really begs the question as to how these things and how these events happen. And so I suppose we can put ourselves in the context of just considering some of the historical information that we've had for a number of years now, which describe what we call intrinsic contamination and extrinsic contamination as a means by which Cronobacter species can contaminate powdered infant formula. So the intrinsic contamination is a contamination event that usually happens during production. And so here you just have a very schematic outline of a production process. And on the left-hand side of my slide, some of the considerations in terms of the phenotypic or challenges or threats that are posed to Cronobacter and which it must overcome in order to survive in this environment. Then you have extrinsic contamination, which happens outside of the production environment, and probably more to do with the home or hospitals where you have caregivers preparing powdered infant formula for young infants to be consumed later on. And when you think about that consumption, you think about a child which perhaps doesn't really look like this type of a healthy looking baby, but where the ingestion of contaminated powder, reconstituted powdered infant formula causes initially a disturbance at the intestinal epithelial surface, where you may get a necrotizing enterocolitis. And then if the bacterium finds its way further and deeper into the system, you may get a bloodstream infection. And if it presents itself at the blood-brain barrier, you then are at risk, or the baby is at risk of developing meningitis. And in many of the severe cases, meningitis is the outcome, and it's often fatal. And again, historically, when you look back at the recorded cases of infections in infants, this was a paper produced in 2006 by Anna Bone from CDC. And Anna did a trawl back through the case reports at CDC at the time to try to highlight any of them that may have been linked to the consumption of powdered infant formula, contaminated powdered infant formula. And of the 46 cases that were identified at that time, a number of them were associated with meningitis, a number of them associated with bacteremia, just as I've said, one associated with the urinary tract infections. And in several of these cases, there was examples of epidemiological links, whereby the bacterium was cultured from the ill infant, and it turned out to be the same organism that was then subsequently cultured from the contaminated batch of powdered infant formula that was consumed by the infant. So this was providing epidemiological evidence, if you like, of a link between the food chain and the contamination of the infections in the infants. Subsequent to that, another study, again published from CDC in Emerging Infectious Diseases, looked at a number of different age groups over years and anatomical sites from which the organism could be cultured. And you can see here that the bacterium can be cultured from a whole host of different anatomical sites, including cerebrospinal fluid, blood, and so on, as I've mentioned 2 slides back. And when you look then at the age distribution of individuals who are at risk here, predominantly it's those at the extremities of age, either the very young, in this case, vulnerable infants that may be presented with contaminated powdered infant formula, or people like myself on the other extreme, the older age group, whereby these individuals may require medicated type foods because of difficulties with their swallowing mechanism. And I think based on what we now know about the epidemiology here, certainly we'll probably see a larger number of cases reported from groups coming on the right-hand side of my slide here, so the older population, as opposed to those in the younger population on the left-hand side of my slide. But nevertheless, the infections in infants is much more severe. So I suppose if we were to try and draw some of this together and say, well, what do we actually know about Cronobacter infections at the moment? Well, I've said something already about the types of clinical presentations or symptoms, so necrotizing enterocolitis, infections in the bloodstream, or meningitis. For children that may survive these infections, there's the risk of long-term complications, such as those illustrated here. But there's still a lot that we don't know about this bacterium. For example, we're not entirely sure what the infectious doses actually is, although we do have some idea what that might be, or the period or duration of illness. But one thing we do know about is that powdered infant formula is one of those foods that potentially presents this problem, or from which this problem can be presented. And similarly, I suppose we could add here medicated foods for older individuals. So that in turn begs the question further as to where Cronobacter actually comes from. So way back in the early days of studies being reported, Carl Iverson and Steve Forsyth, Steve is one of the panelists, presented this hypothesis that perhaps this organism could be found in the natural environment associated with plants. And I think there's probably a lot of truth in that, in the sense that this paper here, which was then subsequently published in 2009 in Research in Microbiology, very elegantly showed the fact that Cronobacter sakazaki can colonize the roots of plants. So you can see here some of these very nice confocal microscopic images showing the presence of the bacterium actually embedded in the plant cells itself. Again, subsequent to this study, and very recently, in fact, in the last couple of days, a paper that appeared in Microorganisms, the journal Microorganisms, took up this theme of plant-associated foods and indeed described some very nice, elegant comparative genomics, which showed similarities between isolates that are recovered from plant origin foods and similarities with other isolates that have been known to cause clinical infections. So there may be a link here which is borne out by the epidemiological and genetic evidence. In terms of what actually is or what constitutes Cronobacter species, we have to go back again to the early 2000s. And this is one of the pieces of evidence which suggested at the time that Enterobacter sakazaki was actually a conglomeration of different species. And I suppose it's fair to say that when we think about the genus Enterobacter, many of the bacteriological taxonomists might regard that as a type of parking site, if you like, where when we don't know what a bacterium is, we simply call it Enterobacter something and just park it there until we get more intelligence. And so when you think about this historically, what you're looking at here on the left-hand side is a genetic tree whereby a comparison was done using the full-length 16S ribosomal RNA genes from a large collection of different Enterobacter sakazaki from different sources. And clearly you can see here a delineation into groups, a number of different groups, 4 in this case, which essentially resulted in the conversion or the updating of the name Enterobacter sakazaki to Cronobacter species. And at that time, 6 of the now 7 recognized species were identified, and these were clearly differentiated from other Enterobacter species, which can sometimes be found in the very same environments, but which are not at this time known to be pathogenic. Again, that taxonomic delineation and the updating of the genus name was confirmed by SNP analysis, again looking at whole genome sequences of a large number of strains. And clearly you can see here from my slide the differentiation between those species that are clustered within the Cronobacter genus versus other non-Cronobacter species that can now be separated from these. Again, there's some very well-established phenotypic methods that can be used to draw these distinctions, and this slide just illustrates some of those and some of the types of reactions, biochemical reactions, that can be used to distinguish between Cronobacter species and non-Cronobacter species, and that can provide that very fine distinction between Cronobacter species and those Enterobacters that I mentioned a few moments ago that can sometimes be found in the same environment, but which are not pathogenic as far as we know. So this brings us then to this whole question of adaptation. And again, from work done by several people in the Cronobacter community, including Steve Forsyth and others, there was a definition of what they called a clinically important sequence type called ST4 some time ago. So a large number of these Cronobacter Sakazaki strains that were being reported in clinical infections, when they were subtyped using multi-locus sequence typing, ST4 seemed to be a predominant type. Other sequence types are well known, for example, ST1, as I'm showing you here. But what this slide shows you is a comparison between isolates that are defined as ST1 on the top panels versus isolates which are defined as ST4, coming from either PIF origin, so powdered infant formula origin, or from clinical origin. And what I think this shows you very nicely is that bacteria appear to adapt into specific ecological niches. And our understanding of that key principle is critical to informing our ability to solve this issue of contamination in a production environment. So to that extent, if you look at this very early study, which looks at the contamination of a powdered infant formula production site, you can see here that over a period of a year, swab samples and powdered samples were taken from different locations within this particular factory. And at the time, they were identified as Cronobacter Sakazaki, as is the case here. And then each one of these individual isolates was then fingerprinted using pulse field gel electrophoresis. So this pulse field gel electrophoresis protocol became part of the PulseNet system at the time, which is now moving over to the use of whole genome sequencing to do exactly the same thing here. But I think the principle here is important, and the principle here is the fact that you can see here that where we had individual pulsotypes designated by these colored dots in a very simplistic way, you could map these colored dots back to the locations from which those isolates were cultured, and you can clearly get a picture of the contamination within this particular environment, and not alone that, but the transmission routes also from that environment. Over a period of time, if you follow this out for a longer time frame, you can begin to get a picture of the colonization dynamics within that environment. And again, using pulse field gel electrophoresis, or today, if we were to do this study, we'd use whole genome sequencing. But again, to illustrate the point here, you can clearly see that there appears to be a prevalent or persistent strain that is being picked up constantly within this environment. And we can see this by way of the conservation across the pulsotypes. So this goes back to that adaptive notion that I mentioned earlier on, and this importance of trying to understand how it is that these strains can adapt to these different environments that may make them a potential food safety risk. Again, when you look at this particular isolate, and this is now known as Cronobacter Sakazaki SP291, and you compare its tolerance to desiccation over time, the turquoise profile, or the turquoise curve that you see here at the top of my graph over time shows you that this strain persists and can tolerate desiccation much better than several other Cronobacter species, in addition to non-Cronobacter isolates that were also examined in this particular study. And indeed, if you measure its thermal tolerance to dry heat, you see here in the blue, right in the middle of my slide, that SP291 is highly tolerant to dry heat at 60 degrees C for a period of over 2 hours, in comparison to other strains of Cronobacter Sakazaki, or indeed isolates of Salmonella, which were involved in another type of outbreak much earlier on. So of course, in order to try to tease apart what's going on here in this particular organism, it begs the question as to how is Cronobacter sakazaki sp291 responding to this selective pressure, if you like, by way of this dry heat tolerance? In order to answer that question, we had to go and determine the whole genome sequence of this particular strain. And this isolate turned out to have a chromosome of about 4.3 megabases, in addition to 3 plasmids that were contained within the bacterium. And to address the question that I've just posed, we turned to a technique called RNA sequencing. And this technique was designed to try to give us a picture of the transcriptome when we make a comparison between the bacterium grown to early stationary phase versus when the bacterium is desiccated onto a stainless steel surface. So there's a growth curve that you can see here on the top right-hand side of my slide, which shows you the typical shape of a bacterial growth curve. The pink arrow illustrates the early stationary phase time point at which the culture was selected for the experiment. And the inset figure that you can see is a desiccation curve, which shows you that by way of the red line, when Cronobacter sakazaki sp291 is dried down in powdered infant formula, it tends to survive for a very long period of time. And again, this is really confirming what other researchers have reported up to that point. So then when you turn to the RNA-seq experiment itself, it requires the isolation of RNA from the culture at early stationary phase. Similarly, the isolation of RNA from the culture in the desiccated phase, and then the subsequent deep level sequencing of those RNA transcripts and the mapping of those transcripts back onto the genome as I just described in the previous slide. So what we're looking for here is a picture from the transcriptome of genes that are highly upregulated versus genes that are highly downregulated versus genes that don't appear to signal at all. And in the pie chart on the bottom right-hand side of my slide here, you get that summary picture whereby the green illustrates genes that are upregulated, the pink illustrates those genes that are downregulated, and the soft orange color shows you the numbers of genes where there's no change in gene expression. We can illustrate this based on the classification of orthologous genes by way of genes involved in amino acid metabolism, and you can see the signaling there. Similarly with respect to carbohydrate metabolism, and so on. But if we look more specifically at genes involved in stress responses, and in particular genes that are involved in osmotic functionality, we can begin to see a very interesting picture which emerges, which tells us that when you take that bacterium from an early stationary phase liquid culture and you grow it or you desiccate it onto a stainless steel surface, the bacterium suffers a very deep osmotic shock, and it's going to respond in order to attempt to protect itself by way of upregulating genes that help it to counteract this osmotic stress. So this is what this slide is actually telling you. And this is just showing it to you from the point of view of the DNA strand itself, where many of these genes are located. And clearly here you can see two operons, the betaine metabolism operon on the left-hand side, the proline metabolism operon on the right-hand side. And you're seeing here a comparison of the RNA-based transcriptome at early stationary phase in green versus what happens in red, which describes the desiccation phase. And clearly you can see that the BET-I, BET-A, and the BET-B genes are switched off during the early stationary phase, but when the culture is then desiccated, you see these genes kick into life by way of the wavy line that you see in red at the left-hand side of my slide. Similarly with respect to proline metabolism. We can confirm these observations by standard quantitative PCR, which validates what we see in the RNA-seq experiment. So from the RNA-seq experiment, we begin to get a picture of what the bacterium is doing when it's in this kind of desiccated tolerance state, if you like. And one of the most important genes, or one of the most important metabolic pathways that appears to be functioning is one involving trehalose metabolism. And this is just a simple schematic on the top of my slide here to show you what's happening by way of trehalose when it's imported into the bacterial cell, and then it's converted by way of several biochemical transformations, if I can put it like that. So if we knock out genes involved in the metabolism of trehalose, then the question is, does that mean we can make this bacterium much more susceptible to survival during desiccation? And in fact, that's exactly what happens. So if you look at the right-hand side of my slide, the desiccation curves again here show you the original strain SP291 in blue and its desiccation curve versus the isogenic equivalent of SP291 in red. And clearly you can see here that there's a difference between the survival of the wild type SP291 strain in blue and the mutant where it's lacking this trehalose functionality in red. So we're beginning to now understand what's happening to the bacterium once it goes into a desiccated and tolerant condition. So the final question here is this a general observation for all Cronobacter sakazaki strains? And in particular, going back to what I said in terms of the adaptive nature of different types of Cronobacter sakazaki. So we redid the experiment again, but this time we put in a second strain, which is this ATCC29544, which is a clinical isolate. And we ran through exactly the same experiment as I've just described before. And really of note here is the bottom right-hand side of my slide, which shows you again the desiccation curve. So the green line you see here is the clinical isolate 29544. The red isolate is SP291, which, as I mentioned, we've picked up several years before in a powdered infant formula production environment. So it appears that there is a difference between the ability of the clinical isolate 29544 to survive desiccation versus what we see with respect to SP291. Again, we can look at the RNA-seq data, and we can see here broadly that there are some differences when you look at genes that are signaling during what we call the primary response, which is mainly where potassium ions are transported into the cell, versus genes which are signaling during the secondary response, which is where we see trehalose and many of those types of transporter systems responsible for osmoprotection transportation into the bacterium also signaling. And similarly, on the right-hand side, there's a difference we can see here with respect to differentially regulated genes that function as transcriptional factors. So it appears that the networking, if you like, in the SP291 genome is a little bit different from what we see in the 29544 genome. And this is borne out by the final slide to describe this experiment, which just shows you the magnitude of expression of some of these important genes that are signaling during desiccation in SP291, which is a bacterium of powdered infant formula origin, versus 29544, which again is a bacterium of clinical origin. So that adaptive nature appears to be coming through here in some of the results from these experiments. Now, there's been a lot of discussion by powdered infant formula companies and other food companies about some applications of sequencing technologies in terms of trying to help them to improve food safety measures within their production sites. And there's several ways in which you can approach this. I've just been describing a single bacterium focused approach where we're interested in identifying the causative agent. But what happens if we pull back and we take a look at the totality of the environment that we're trying to describe? And here there are several different sequencing approaches that you can take, including the use of 16S ribosomal RNA-based sequencing. I mentioned that earlier on, which will identify bacteria, ITS sequencing, which will identify fungi, or a more general approach, which is called shotgun sequencing, which will identify everything in addition to genes associated with some types of functional categories that we might be interested in. So it was of interest to see, could we apply this to two different powdered infant formula production sites? And these are just schematics showing you both of those production sites, Factory M and Factory W. Factory M was a pharmaceutical grade powdered infant formula production site. Factory W was an old food grade environment. And what we were trying to do here was take samples from these environments over time and to carry out both 16S ribosomal RNA sequencing and shotgun sequencing to try to describe the nature of the microbiota that's present, and particularly to try to recognize signals when these things changed or when these microbiota compositions changed, and then try to tease out why they changed. So when we carried out this analysis, sure enough, we could identify a core microbiome in both of these locations. And the core microbiome is summarized for you here on the bottom right-hand side of my slide. And so now we begin to understand the nature of the bacterial microbiota, if I can put it like that, that seems to colonize these different production sites. And by and large, the core microbiome was very similar in both places. But there were differences. There were some clear differences depending on whether you were in factory W or factory M as to the makeup of the constituents within the microbiomes that we were describing using both 16S ribosomal RNA sequencing, which is what this slide is showing you, and also shotgun sequencing, which I have not included. And when we begin to try to relate that back to events that actually happen in these production sites, we begin to get clues as to what may be the cause of some of these perturbations that we were seeing in our sequencing with respect to different locations within this particular production site. So here you can see the medium care area, the low care area, and the high care area, and the effects of movement of individuals, routine cleaning, and so on the nature of the microbiome as it was established using 16S ribosomal RNA sequencing. This may be something that will come up in the panel discussion later on, so I think I'll not say any more for now, and instead just go to the conclusions. So I think based on my presentation, I hope I've managed to convince you that understanding the microbial ecology of a food production facility is very important, and it's important in terms of trying to help you to manage food safety risk. It's also important to try to differentiate persistent from non-persistent isolates, and therefore it gives you a chance to act in terms of trying to prevent outbreaks that might occur if such actions were not taken. And then I think as many food companies are looking at the moment, the use of whole genome sequencing and trying to evaluate that in the context of how it might be applied to improve their food safety countermeasures, I think is something that's going to continue in terms of the discussion. But I am convinced, and perhaps you might say I am convinced, that using sequencing will provide us with an ability to identify novel biomarkers, which might then subsequently aid in a more rapid detection and subsequent risk reduction. And with that, I'll thank you very much for your attention. So thank you.
Thanks a lot, Seamus, for the excellent introduction and insights already. There are already questions coming in, so let me start with the first one. Our elderly parents are encouraged to consume protein drinks. Do any panelists have published data on Cronobacter on protein drinks or protein bars? And maybe to extend this to the question, what do the panelists think about the importance or significance of plant-based foods as an origin for Cronobacter? Or vehicle, not origin, it's the wrong word, but vehicle. So I don't know, Seamus, do you want to start, or Stephen? Or?
Well, I'm happy to attempt to kick off the discussion, and I'm sure others will join in. I'm not aware of any specific studies that describe the nature of the food matrix that was in your question, Annette. But nevertheless, having said that, there would appear to be foods where there's a low moisture content, and where there is a low moisture content, you have to kind of wonder whether or not there's a risk of some kind with respect to Cronobacter, but also in this context, salmonella. And I didn't mention salmonella at all in any of my comments. It's equally valid for what I'm saying here. So that's the first thing. Others may have an opinion on that as well, and I'm sure they'll chime in. The second part of your question, if I can attempt that also, I think, as I've said, you know, historically work from Steve Forsyth's group and others have highlighted the importance of plants as a potential source. So in many of the formulations that you see produced by the manufacturers, there's sometimes the use of spices as part of the formulation. And so one has to wonder whether that's a potential ingress route for contamination during the process. And again, as I've mentioned during the presentation, there is evidence which has now been just produced, which shows that Cronobacter isolates that are cultured from plant origin foods show similarities to many of the strains that are associated with clinical cases of infection. So perhaps we need to start thinking about widening our surveillance purview, if you like, in terms of bringing in plant origin food into the paradigm.
Annette, if I could just come in. I think one of the issues, it's a good point raised in that question. One of the issues I think at the moment is in routine screening of foods that will be done, nobody outside of the infant feed area is probably monitoring for Cronobacter. There's a complete lack of knowledge about where it is because nobody looks for it. And I remember a few years ago working with one company, and they happened on a confectionery product, not designed for infants or children, just a general confectionery product, them getting a very high Enterobacteriaceae count. Normally, they would shrug their shoulders and say, yeah, high entero count, what do I do? For some reason, they decided to get it identified, and they identified Cronobacter sakazaki, which gave them another problem to deal with. But I think it may be a hidden issue that we don't realize is there because nobody's actually doing testing specifically for the organism outside of the infant feed area.
Yeah. Thank you. So what I'm hearing that we don't have really data on the prevalence of Cronobacter in the different foods or in the different plant-based foods, apart from the infant formula where it's been actively looked at.
We do have data, sorry, Steve Forsyth, we do have data with regard to, as Shay pointing out, about the organism in plant products such as wheat and rice. We did a study, total about 20,000 samples that we published last year. And when you look at things like rice and cereals, you're talking about 10 to 25% positive for Cronobacter. And rice, sometimes different countries vary in what they're using as infant formula. And it's one of the things of this discussion to be a little bit careful as to what we're calling infant formula with regard to the target age being less than 6 months or over 6 months, which for some of us would be called follow-on formula because it's not the sole source of nutrition. So it is quite common in plant-based materials. I don't know about plant-based foods and protein drinks as such. I only know of one case where they suspect in an adult there was an infection through the food, which was in China. But that's the only suspect case I'm aware of. So I think it's one of those organisms that if you look, you'll find it, but we're primarily concerned with, in a neonate, they are highly susceptible, as Shay's pointing out, and that's where the focus is, to me correctly, in the food industry. That's the risk because of the effect. But the organism is very abundant in plant material, and therefore in starch. I mean, we used to do class practicals with tea bags. If you want a collection of Cronobacter, use tea bags, cake mixes, things like that. You'll find the organism quite abundantly.
Thank you, Stephen. Maybe we move to the next question because also time is running. So the next question is Cronobacter also found in wet processing environment or is greatly specific to dry environments? One of the illustrations shared showed the isolates were not found on the wet site.
I think that might refer, Annette, to one of my slides. Yeah. You should not get drawn into that actually, because we simply didn't look on the wet site. But if you talk to people involved in powdered infant formula production, many of these production environments, they will try to run their factories in a dry condition. And the reason they're trying to do this is to try to maintain the EB count as low as possible. So what Roy said earlier on is very important here. So the EB count, if they can maintain it very low, it may go some way to reducing the risk of Cronobacter. When you start to wet that environment again, let's say at a maintenance intervention or a maintenance shutdown, and you start to introduce water, you start to get an increased EB count. And if Cronobacter is there, that can also start to grow also. So in the slide that you're referring to, we didn't look at that wet side for those reasons. We only looked at the dry side of the production site.
Maybe that links into environmental monitoring as an important preventive control. Then are there some recommendations from the panelists how to establish the how to say, an effective environmental monitoring for Cronobacter? Francois?
Yes, if I may, yes, thank you, Annette. Indeed, when you consider Cronobacter as a pathogen of concern, this is also asked, if you look closely in the EU regulation, you should focus on these specific organisms if you do produce for neonates in your facilities, and particularly for low moisture food. That's what we mentioned in the EC Europe publication. And be aware that you need to focus on the pathogen of concern. It's not enough to focus on the hygiene indicators. It's indeed interesting because if it's hygienic, it should be safe, but it's not because it's hygienic that it's safe. And that's something you see very embedded in the EU regulation. In the first version of the 2073-2005 regulation, they mentioned that you should look for Enterobacteriaceae for infant production. Then they make a modification 2 years after in the 1441, when they mentioned that there is no correlation between the presence of Enterobacteriaceae and the detection of Salmonella, and it's up to the food business operators to show clear correlation between presence of Cronobacter and presence and enumeration of Enterobacteriaceae. True reason for this, the limit of detection is not the same. You've got enumeration of one side, you've got detection, non-detection on another. The ecology is not exactly the same. As mentioned by Seamus, Enterobacteriaceae is a broad family, and then the ecology is very broad. Not all Enterobacteriaceae are the same, and there are some specificities from Cronobacter as well as there are specificities to Salmonella. Therefore, in your processing environment, if you do produce at some point production for less than 6 months, even if it's only a part of the production, you should have Cronobacter as a pathogen of concern monitored in your environment. Then where to focus, it's basically the same as Salmonella. You should look for harborage niches where potentially you've got organic matter as a contaminant, where water presence that can help the survival. What is relevant for Cronobacter and Salmonella, they survive dry condition, and therefore they will be pathogen of concern for dry environment. It's not that clear for wet environment. First, we don't have a lot of wet environment with infant formulation. Luckily, we stopped this, and as well, per se, they are not a very good competitors in this type of ecology.
Yeah, thank you. Thanks for the insight. And then, so already the next question's popping up. Seamus, you said you don't know the infectious dose, but we have some idea. Can you elaborate on this? Respective infectious dose, what is known or what do we know?
Yeah, so some of that information in it would have come from studies done some years ago, looking at various kinds of models to try to estimate what the dose might likely be. So that's really the only information that I think we have at this stage, unless any of the other panelists have more recent data.
Hi, Annette?
Yeah.
Sure. I was going to follow up on that. The issue of infectious dose is always an awkward one, because let's pretend you say 1,000. It doesn't mean that 999 is safe. So let's just be a bit cautious with regard to precise number. And I'm sure Shay, agrees with that. We try to. It helps to teach students about an organism, but one has to be careful. The thing that I would bring out is, most of these infections are in premature babies, and therefore they don't feed from a bottle. They have a nasogastric tube, which may be in place for days or months. In some countries, they fill up like a syringe, almost like 100 mils of the rehydrated formula, and they slowly pump that into the baby. That happens in America and in France. So, given that syringe is going to be at ambient temperature in a neonatal intensive care unit, gives an enormous opportunity for whatever that initial number is in the infant formula to multiply up over time. You do get biofilms in enteral feeding tubes. We've pulled out meningitis E. coli K1 from the feeding tubes of 11 babies on different feeding regimes. We pulled out Cronobacter sakazaki from feeding tubes of babies not fed infant formula. Because bear in mind, the Cronobacter melanaticus type strain came from a breast abscess. So it's not just sakazaki that can cause an infection. Malnaticus can as well. So although that infectious dose may be done maybe in animal trials, the situation when you're feeding a neonate can vary between country, and you have the opportunity of multiplication in the feeding tube, which may be in place 37 degrees. And we count Enterobacter easily up to 1 million in the residue liquid inside the feeding tube. So that's why I just want to pull in, how you feed a baby is different to how an adult eats food.
Yeah, thank you, Stephen. That's a very important point. Marcel, you want to comment?
Yeah, I would also like to comment on the infectious dose. That as a risk assessor, often we see it the other way around. It's not an infective dose, but it is a probability of every Cronobacter to cause a disease, and that is the other way around. And that is also a little bit linking it to what Stephen said. Things can happen, of course, after preparation or in the tubes and so on, but also in the presentation of CMUS. We have seen the process of the organism going further and further in the body, and that is every time, you could say, a probability. So I consider that the infective dose is 1. So one organism can make a baby ill, but it has a low probability because it needs to go through all these steps. And it can be, for instance, that the probability is 1 in 1,000. And then it seems that there is some sort of infective dose, but it is every time a probability. And that is very relevant if you look at the food safety management, because if you have 100,000 cans where in every one of them there is 1 Cronobacter, you get a lot of babies getting ill. Well, if we would consider what Steve was saying, that it would be wrong to say it's, for instance, 1,000 and 999 will not get someone ill. That's a little bit the same concept, that if you have a lot of exposures of one organism and you have every time a small probability, you still get ill babies. And we see now in those outbreaks that there are often only 4 babies or so that get ill from a large contaminated batch with very few numbers of organisms.
Mm-hmm. Thank you, Marcel. And here's another very interesting question related to, has research identified any inhibitory bacteria that can be used to control Cronobacter in factory environments? Anyone has an opinion on that?
So there are a lot of projects in the dairy industry with bioprotection in the processing environment. The issue we have is it works in the labs, definitely. But then you need to have a bacteria that will survive the cleaning procedures as well. And this is where things start to be difficult. The other point is that classically, if you need to inoculate the factory with bioprotective microorganisms, this is something you do with spray, so it's wet, and you try to be dry in the factory. So by design of implementation and by the control measures in place to reduce water in the factory, this is not working. Bioprotection against spoilage or pathogen of concern is something we have in wet dairy products, for example, to avoid fungal contamination on the surface of soft type cheeses. But for this type of organisms in the dry environment with very competitive conditions, this is for the moment, this is what the few we have is not working, and the implementation will be very, very complicated. So this is not where we seem to go. Have in mind that bioprotection per se is not seen as a huge barrier of control measures. It's one additional one. It's helping a bit, but it's not making it all. And already by the FAO work done on this topic, this is something that comes on top of all the other control measures, but it will not be able to replace the existing control measures.
Yeah. So it can be an additional measure if it would be established, but it will certainly not work as the only measure.
No.
So to that extent, there's also a question on the thought. So what are the thoughts on the driving forces that cause environmental Cronobacter to become to and belong to food, clinical, or even environmental persistor categories? So basically, what is the main driver that makes Cronobacter? So, I mean, Seamus, you outlined already some of the characteristics. Yeah, we know also Salmonella is a very good survivor in dry environments. So how do the both relate maybe to each other? Is one better than the other in terms of survival?
Yeah, a very good question, Annette, and I think we only have part of the answer to this question. I mean, in terms of comparisons between what Cronobacter does when you desiccate it versus what Salmonella does when you desiccate it, there are similarities between the two in terms of their immediate response to withdrawing water and creating that kind of osmotic selective pressure that I mentioned. But there are likely to be subtle differences too that may, alter how one does it maybe better versus how another one does it maybe a lot worse. We don't know yet. And I think these are only early studies that provide initial clues and indications as to what's actually happening. There are a whole bunch of other genes, for example, in the RNA-seq study that we didn't look at. I mean, we could spend the rest of our careers looking at this particular transcriptome to try to tease out what's going on. But at least we get some insights into potential targets that we may be able to look at in order to try to make that organism much more susceptible and to try to address what Francois is saying in terms of trying to reduce the colonization potential.
If I may add to the point of Seamus, there is no one single explanation for everything, because every factory by design is different from its hygienic design, from the good manufacturing or bad manufacturing practices in place and the cleaning practices. And this is where you need to work case by case, because each drying tower, or if each chocolate factory, to speak about other outbreaks going on the topic, has different house pets or mother-in-law, and there is an explanation behind. And this is where case by case, as mentioned by Seamus, it's very important to type and characterize your isolate to understand precisely in your case what's going on. To go back to the wet environment with Listeria monocytogenes, there is an interesting publication from Safe Food 360 in Ireland who showed that there was a huge use of quaternary ammonium compounds for many years on Listeria, but at the same time in clinical isolates, there was a resistance to basalconium. And this is how you made the link. You use quack to clean in the food processing environment, and you end up with benzalkonium resistant Listeria as clinical cases. You've got the same in the Netherlands. Does it mean that quack does it all? No, you've got also iron efflux, you've got the different pumps against electrons. So there are many mechanical back in place, and every case is specific, and therefore for each factory, characterize, you isolate, work with academia to understand precisely in your case what's going on, and then you can have tailor tape control measures. But one size fits all solution, you won't get it. The only one size fits all you have is remove the water, remove the water, remove the water.
I think, and the other thing, and Seamus made a very good impression of that with his talk, is that it's very easy to think of one particular microorganism, Cronobacter, as being universally the same. They're not. And Seamus showed that in his talk with the clinical isolate versus the industrial isolate and how different they are. And I think you've got to think when you get into a food production environment, which, as Francois said, can be all different. You've got an ecological structure of microorganisms that may select for particular strains that fit that particular microbial community, that particular ecosystem. And that may drive particular microorganisms to become prevalent, and it might cause particular strains to become prevalent. So it's quite clear that you will get different strains evolving and becoming prevalent in certain environments because they're all different. And I think it's too easy to think of them all being the same when in fact you've got a continuum of abilities to withstand environmental conditions, and they will simply fit in and adapt to a particular system that they go into.
If I can just add to that, I think that's a very nice overview, Roy, of what is kind of happening at a population level. And to try to tease that apart and to try to understand that population structure is where these new sequencing technologies may prove to be very effective. And I absolutely agree with what Roy has said. Some bacteria may actually shape the nature of the ecological environment in which Cronobacter is found, causing it either to survive very well or not survive very well. But we don't know the answer to those things yet. And as Francois said, we do have some theoretical, some experimental evidence, I should say, that when we conduct these types of microbiota analysis, we can see glimpses of potential biocontrol measures that are naturally occurring. But how we actually deploy those then subsequently is another question, for the very reasons that Francois said.
Yeah, thank you. So it seems that it's a very still open and heterogeneous field, and there doesn't seem to be like one solution fits all at this moment in time. So, and because our time is up, but here's an interesting question to all of you. What would you think or recommend for powdered infant formulas factories of the future that has not been widely applied and could be applied to address Cronobacter and Salmonella because of their survival in this kind of environment? Is there any recommendation you want to provide to the audience? Or best practices you could think of at this moment in time?
I'm going to like dodge answering that in a sense, because I think in general, millions of babies are fed safely every year by infant formula. So, you know, the factor of keeping the moisture low, you know, we know it's there. It's ensuring it's implemented is the key thing. So I think the structure is already there. I think millions of babies are fed safely every year using powdered infant formula and as well as by breast milk. So I'm not sure that we need a magic wand as such that we haven't thought of before. I think, you know, the principles that we've discussed this afternoon, one needs to ensure that they are implemented 24 hours, 7 days of the week.
Thank you, Stephen. Marcel, last comment?
Yeah, so one thing what we could also do is that we focus more, you could say, on the risk groups and the specific foods. That's if we also for the elderly, for instance, if it's really medicinal food, for the elderly, Seamus was already saying he was going to the elderly, but he's very healthy. So I don't think that he will ever be infected by Cronobacter. But as soon as you are an almost terminal cancer patient or so, then you become susceptible. Those people need medicinal food, and there you have to pay much more attention to the safety of the product used. And that is the same for the babies that are in the intensive care units, and where you have those problems with the tubing and so on that Stephen was mentioning. There you need to put much more effort, because there is really the problem on the two extremes of the elderly and the very young. And there we could more specifically have specific food where we pay much more attention, making it much more expensive. But then you don't, we are a little bit shooting with a cannon on a fly or on a mosquito. That might also be a direction.
Yeah.
If I may add to the comment of Stephen, yeah, indeed we are recently some incident, but let's not forget that the vast majority of the food is well done. One of the issues with Cronobacter and as well Salmonella is that it comes from the processing environment, so it's important to focus on having efficient hygienic design for all what it's tubes, the milk tower whatsoever. But something very important, the premises, the building, and please a nice roof, because roof leak is really a killer, and that's really something not underestimated. I prefer to have a 20 or 25 years old towers in a nice building rather than the state-of-the-art towers in a not so well designed building. I know which one will fail first, so that's really something to have in mind. There is a lot of investment done in the drying tower to have a specific design. Really, that's a must indeed. Do not forget the building, because I have visited some towers which were very nice, but the floors was within the buildings, and it was a ticking bomb before. And it will be a ticking bomb before problems occur. In the case of the Michigan factory from Abbott, it was flooded. It's right closed right now because it was flooded. So really, the place of the building, the design of the building was not the best. Because that's the issue is not with the tower per se for the right month. The issue is with the location and the design of the building, which can be flooded. This is a major issue, and do not omit this one.
Thank you, Francois. I think with that, I would like to thank all panelists here. And all participants, we are slightly over time, I see, but it was really worth it. It was an interesting discussion. I think what came out of it, just to summarize, there is no like one solution fits all, but we need to stay vigilant. We need to look what is our intended use, we need to look at our risk analysis, risk assessments, and pay attention to all the little things that give the microbes a niche to grow and yeah, getting us into trouble. Again, a note on all the questions. There were more questions being asked. We will make sure they all get answered. We will get back in writing and link with IFP. And with that, I really like to thank all the panelists for their contribution, for their time, and especially IFP for making this webinar possible on such a short notice, and for ILSI in supporting us in arranging it. Thank you, everyone here.
And Annette, for sharing.
Thank you. That
Great.
concludes today's webinar. We would like to thank all the panelists for today's discussion. This webinar is being recorded and will be available for access by IAFP members at www.foodprotection.org within 1 week. Thank you, everybody, and have a nice day.
Thank you, everyone.
Bye-bye. Take care.
Bye now.
And
Ciao
stay healthy,
tutti.
everyone.